Hello, and welcome to the FDA public meeting using methods from PFDD Guidance 1 and Guidance 2 as tools for including patient experience data in clinical trials, who to ask and how to ask. My name is Shannon Cole, and I'm a member of the patient-focused drug development staff in the Center for Drug Evaluation and Research. I will be facilitating the meeting today. This meeting is the first in a series of 2 public meetings. The second public meeting will take place on July 25th from 11 a.m. to 1 p.m. Eastern Time. Patient-focused drug development, or PFDD, is a systematic approach to help ensure that patients' experiences, perspectives, needs, and priorities are captured and meaningfully incorporated into drug development and evaluation. As experts in what it is like to live with their condition, patients are uniquely positioned to inform the understanding of the therapeutic context for drug development and evaluation. FDA is developing a series of 4 methodological PFDD guidance documents to address, in a stepwise manner, how stakeholders can collect and submit patient experience data and other relevant information from patients and caregivers for medical product development and regulatory decision-making. This series of guidance documents is intended to facilitate the advancement and use of systematic approaches to collect and use robust and meaningful patient and caregiver input that can better inform medical product development and regulatory decision-making. These guidances are part of FDA's PFDD efforts in accordance with the 21st Century Cures Act and the Food and Drug Administration Reauthorization Act of 2017, Title I. The first 2 guidance documents in this series have been finalized. FDA is convening this series of 2 public meetings to share lessons learned and best practices based on use of these first 2 guidance documents, and to hear from stakeholders about how they have used these guidance documents as tools to advance their inclusion of the patient voice in drug development. During the meeting today, speakers and participants will discuss a range of issues and best practices related to collecting patient input, selecting data collection methods, and developing sampling plans and strategies. External stakeholders will present examples of how they have used the guidance documents as tools to inform their collection of patient data. Next slide, please. Here's our agenda for today. This meeting will run from 11 a.m. to 1 p.m. Eastern Time. It will be recorded and posted online. This meeting and the second meeting in this series of meetings have an associated public docket in which interested stakeholders are invited to share examples of how they have used the PFDD methodological guidance series to advance the inclusion of the patient voice in the drug development process. The docket is open to the public and will close on September 23rd, 2022. More information on the second meeting in this series and a link to the public docket will be provided later during this meeting. For today, the next item on the agenda is opening remarks, which will be followed by an overview of PFDD. The rest of the meeting will consist of 3 sessions and a clinical regulatory perspective presentation. The first session will focus on research methods to identify and understand what matters to patients, where we will hear from FDA staff members. The second session will focus on ideas and practice, where we will hear from representatives from industry and patient organizations. An FDA staff member will provide a presentation highlighting the experiences of a clinical reviewer within PFDD efforts. Finally, the third session will be a question and answer session where stakeholders are invited to use the Q&A feature in Zoom to ask questions to the presenters. Now I will turn it over to Dr. Teresa Mullen, the Associate Director for Strategic Initiatives, to provide opening remarks.
Thank you, Shannon. And yes, good morning. I am Teresa Mullen, the Associate Director for Strategic Initiatives in the FDA Center for Drugs. And we are very glad that you were able to join us for this PFDD workshop on using methods from PFDD Guidance 1 and Guidance 2 as tools to include the patient experience data in clinical trials. You know, FDA established the patient-focused drug development program, recognizing that patients' experiences living with their disease and the treatments currently available really provides, and also the carers for these patients, gives them a unique perspective, as Shannon was saying, and this can really help inform our regulatory decision-making. It also can inform many other aspects of drug development. Perhaps in recognition of that wide range of information that might be collected from patients and carers, and commensurate with the opportunities, the wide range of opportunities to inform decision-making, the law provides us with a very broad definition for patient experience data. Patient experience data includes, first of all, it has to be collected by any person, including patients, family members, caregivers of patients, patient advocacy organizations, disease research foundations, researchers, and drug manufacturers. And secondly, intended to provide information about patients' experiences with a disease or condition. So this includes the impact of such disease or condition or related therapies or clinical investigations on patients' lives, and patients' preferences with respect to treatments for particular diseases or conditions. But FDA also notes that the quality of that collected patient experience data will really determine the extent to which we can use it in regulatory decision-making. My father was in the building trade, and he used to say, measure twice, cut once. And this is a common expression in that line of work, but I think it's relevant here too, reflecting the importance of good planning and preparation, including a thoughtful approach to measurement and construction of instruments and approaches. If drug developers and others are going to engage patients living with chronic disease and ask them to spend time and provide information about their experiences and with their disease and their preferences related to treatment, FDA would like that to be done with a high quality approach to make good use of patients' time and effort. We would also like the approaches and the methods and tools used to be valid and reliable so that we can use the patient experience data to the maximum extent possible. The PFDD Guidance series under development is intended to support that very important objective. We not only want to issue the guidance series, but we also want to engage patients and sponsors and other stakeholders to hear about their experiences and learnings in applying the guidance. So thank you again for joining us today. We really look forward to the discussion. And with that, I want to turn it over to Robin Bent, who's the director of our patient-focused drug development program. Thanks, Robin.
Great. Thanks so much, Teresa. Thank you, everybody. Welcome to the meeting today. It's really great to be able to speak with you. I'll be building on Shannon's introduction. Next slide, please. So over the next few minutes, I'm going to provide you with an update on the patient-focused drug development series. As we go through the meeting, I ask you to maybe put any questions that you have into the Q&A box, and then we'll answer them during the question and answer session at the end of the meeting. But you certainly don't need to hold your questions until the end. It's just when we'll be able to address them. So next slide, please. So talking a little bit about Guidance 1, this first guidance was, as Shannon mentioned, published in draft in 2018 and was finalized in June of 2020. It discusses sampling methods that can be used when planning a study to collect patient input. It also provides a general overview of the relationship between potential research questions and methods when deciding from whom to get input. And this includes defining the target population and developing the sampling strategy. Next slide, please. Guidance 2 was just finalized in February and discusses methods for eliciting information from individuals identified in Guidance 1. It presents a range of methods and established best research practices to identify what's important to patients with respect to burden of disease, burden of treatment, and the benefits and risks in the management of a patient's disease. These two documents are the tools that we'll be discussing today, so you'll hear a lot about them in just a few minutes. Next slide, please. Guidance 3. So Guidance 3 actually published yesterday, or technically today, but was available yesterday. This guidance is a collaboration between the Center for Drugs, the Center for Biologics, and the Center for Devices. And we hope that you will find it worth the wait. I'd like to take this opportunity to give a huge shout out to everyone who worked on this guidance or had a hand in getting it published. It truly was a massive lift, and a lot of our staff members did this on top of an already exponentially expanded workload because of the pandemic and such. So in case you haven't had a chance to read the guidance all the way through yet, this guidance addresses refining the list of concepts of interest important to patients for measurement. Given that not everything identified as important by patients, caregivers, and clinicians can be addressed by an investigational treatment or be measured, this guidance will address issues related to selecting what to measure for medical product development and identification or development of fit for purpose clinical outcome assessments of importance to patients. Next slide, please. And guidance 4, the fourth guidance in the series, will address topics related to incorporating clinical outcome assessments into endpoints for regulatory decision making. And this includes clinical outcome assessment related endpoint development, defining meaningful within patient core changes, and collection, analysis, and interpretation and submission of data. Next slide, please. So here you'll see a screenshot of the FDA website for guidance 3. You'll see that there is a link to comment online. I will go ahead now. We'll see if I can do this. Drop a link in the chat for everybody in case you're interested. And we do really encourage you to take your time, review the document, and then to provide us with any comments. You can see that the comments were open to comment through September of the end of September. So there's plenty of time to review the document. If possible, when you submit the comments, if you can submit them in a Word format or something like that, it makes it a little bit easier for us to review the comments, but however you submit them, they will be reviewed and considered as we move from draft to final. Next slide, please. And so here on this slide, you'll see 2 resources that we're rolling out, and we're really very excited about them. We kind of realized that as the guidance series is progressing, it's getting a little bit increasingly more technical. And honestly, it kind of needs to be so that the subject matter experts, like social science researchers, psychometricians, statisticians, clinicians, and others really understand what FDA needs to see to be able to use the very valuable information collected in clinical outcome assessments to inform our regulatory decision making. But we know that not everyone has the time or sometimes even the desire to read a lengthy guidance document. So what we've done is to develop a guidance snapshot that's kind of a quick 4-page read that covers the highlights of the recommendations in guidance 3. We've also developed a podcast where you can hear subject matter experts talk about the importance of the guidance document. That podcast is about 11 minutes in length. I'll drop these links into the chat as well. Although the slides with the links will be available kind of following this meeting. And next slide, please. And finally, before turning it over to our presenters, I would like to mention the International Council for Harmonization ICH PFDD reflection paper. The goal of this paper is to take steps to harmonize the approaches, methods, and standards to advance incorporation of the patient perspective in drug development kind of more globally. The goal is to build on existing work and not necessarily reinvent the wheel. The paper has been endorsed by the management committee, revised based on public comment, and is available on the ICH website. We haven't begun work on these guidelines yet, but we expect that it will move forward as resources allow. We're very excited about this as well. So with that, I'd like to thank you for your time, and I look forward to your questions at the end of the meeting. Let me turn back over now to Shannon.
Thank you, Robin, for providing that overview of PFDD. Now we will transition to our first session of the meeting, which focuses on research methods to identify and understand what matters to patients. The objective for this session is to provide an overview of research methods to identify and understand what is important to patients with an emphasis on practical implementation. First, we will hear from Dr. Selena Daniels and Dr. Naomi Noble from the Cedar Division of Clinical Outcome Assessment. They will discuss approaches to collecting patient input and selection of data collection methods. Now I will turn it over to Dr. Daniels and Dr. Noble. Please turn on your cameras and begin your presentation.
Well, hello, everyone. I'm Dr. Naomi Noble. I'm a pediatric psychologist and reviewer with the Division of Clinical Outcome Assessment. And together with my colleague, Dr. Selena Daniels, pharmacist and team leader with the Division of Clinical Outcome Assessment, we'll discuss approaches to collecting patient input and how to select the data collection method. Next slide. A key message of these first two patient-focused drug development guidances is that eliciting what is important to patients can be done using good research practices and methods. And so there are multiple approaches to collecting patient input, but three broad methods are qualitative, quantitative, and mixed methods. Qualitative methods typically are used to obtain a deeper understanding of the patient experience by generating in-depth information from patients in their own words. Quantitative methods involve quantifying the data, so getting numerical data, for example, and applying statistical methods to summarize the patient experiences that have been collected. Mixed methods involve using both qualitative and quantitative approaches in a single program of inquiry to understand the patient experience. And typically in mixed methods, both the qualitative and quantitative findings are combined. So these three research methods can be used to elicit what's important to patients, which may in turn help inform understanding of the disease or condition and clinical trial design. And it might also help generate patient experience data, which can be used to inform the selection of clinical outcome assessments, patient preference information, and benefit risk assessment. Next slide. So within these three broad categories of research methods are a diverse collection of specific approaches that can be taken to elicit the experiences of patients, their caregivers, and other key stakeholders. So as you may see on this slide even, some specific research approaches can be used in more than one way. For example, exit interviews and surveys can be used in both a qualitative or quantitative approach. It's even possible that you might even be able to use exit interviews and surveys as a mixed methods approach as well. And so in the next few slides, I'll give brief examples of these three broad research approaches, and then Dr. Selena Daniels will discuss considerations for selecting patient-focused methodology. Next slide. So as an example of qualitative research methods, just as a reminder, qualitative methods are typically used to obtain a deeper understanding of the patient experience, generating in-depth information from patients in their own words. And so an example here of qualitative research methods is exit interviews, and they were used in a supportive role for product approval. And in this particular example, exit interviews were used with adult patients with carcinoid syndrome, and the public review documents are linked at the bottom of the slide for your reference. A subset of patients from a clinical trial were interviewed at the end of the study prior to unmasking about their symptom priorities and what degree of change was clinically meaningful from their perspective. And as you can see on the slide, patients with carcinoid syndrome described a number of symptoms and impacts and reported what was clinically meaningful for them in terms of symptom reduction. Importantly, patients who experienced a reduction in bowel movements described a sense of freedom from having to go to the bathroom, that they were better able to participate in physical and social activities. And these supportive qualitative exit interview data were considered as one part of the total package of evidence that was considered in the review of this product approval. Next slide. Examples of quantitative methods here. This is an example of survey research methods, and this is a common form of using quantitative data from patients to gather and summarize patient experiences. As many of you may know, the voice of the patient meetings and their subsequent reports are one way to systematically obtain patient and caregiver perspectives on specific diseases and conditions. And this is, to include the most significant symptoms and their impact on daily life. This particular example is from the voice of the patient report for people living with eczema. And the link to the report is at the bottom of this slide. And as part of the meeting, participants responded to a survey question: what are the 3 most problematic eczema symptoms you or the person you care for with eczema has experienced? And the graph that's illustrated here are the responses to this question. And it's a nice example of using survey methods to gather and then represent and illustrate patient experiences. I think one aspect of this graph effectively illustrates the similarities and the differences in eczema symptoms across children and adults. And this is important to capture, especially in conditions that impact people across the developmental age spectrum. And so I think it's important to note, though, too, that while patients who are involved in the voice of the patient meeting can't claim to fully or completely represent all patients with eczema, these voice of the patient meetings and their resulting reports have become an essential resource for offering insight into patient perspectives. Next slide. And then an example here of mixed methods research is really an introduction to these methods. Qualitatively driven sequential design is one approach of mixed methods research. It can also be quantitatively driven, or what's called concurrent or convergent design. In qualitatively driven sequential mixed methods research, the qualitative research is then followed by quantitative evidence. So if, for example, focus groups with patients with diabetes can be used to identify patient experiences, and then a diabetes-specific survey can follow to understand and capture the prevalence. Quantitatively, you can have survey data results that are explored in qualitative interviews to, again, gain a deeper insight into the findings and contextualize them. And a concurrent example would be qualitative exit interviews with patients that are used to contextualize the proportions of treatment responders, for example. So thank you so much for your time and attention here. Next, Dr. Selena Daniels will discuss considerations for which of these methods to use.
Thanks, Naomi. So continuing on, this table is an excerpt from a table in Guidance 1, and it presents some basic distinguishing features of qualitative, quantitative, and mixed method research approaches. Naomi nicely provided a brief overview of these methods, so I won't go into great detail into this slide, but I do want to point out the common research objectives of these methods, as well as how data from these methods are collected. Qualitative methods are generally used for the research objective of describing, understanding, and exploring a phenomenon or a topic of interest, whereas quantitative methods are generally used for the research objective of numerical description, causal explanation, and prediction. Mixed methods are generally used to carry out multiple objectives, which includes the objectives of both qualitative and quantitative methods. With respect to data, qualitative and quantitative methods generate different types of data. To no surprise, qualitative methods produce qualitative data, which is expressed as words or meanings, non-numerical data. And quantitative methods result in quantitative data, which is expressed in numbers. Both are important in gaining different kinds of knowledge. And of course, mixed methods generates both non-numerical data and numerical data. In addition to generating different types of data, qualitative and quantitative methods pursue different approaches to collect and analyze data. Qualitative and quantitative data can be collected using various methods, as Naomi wonderfully laid out. For the context of identifying what is most important to patients, qualitative data can be collected via interviews, including focus groups, observations, open-ended questions, and a survey instrument, just to name some examples. And quantitative data can be collected using survey instruments that include questions with a fixed set of response options, just as an example. I do want to emphasize that it's important to use a data collection method that will help answer your research questions, which brings us to the question, advance please, of which research methods to use to help identify what is most important to patients. For best practice, your research objectives and questions should inform which methodological approaches you use in your research. But taking one step back, even before the development of research questions, when planning research to understand what matters most to patients living with a disease or condition, you should begin with a characterization of the disease and currently available therapies. Prior to conducting studies in patients and/or caregivers, consider conducting literature reviews, consulting with relevant subject matter experts, and utilizing other information sources. All of this can help you develop and determine targeted research questions and to select and decide appropriate methods to identify what matters most to patients regarding their experience with their disease or condition and guide you in meeting your research goals, as well as provide evidence to support your research questions. The key takeaway here is that qualitative methods, quantitative methods, or mixed methods research can be used to identify what is important to patients, and the selection of the method should primarily be guided by the research objectives. Advance, please. However, there are other factors that are important to generally consider when selecting a research group approach, which include 1, research goals or questions to be addressed, which I just touched upon. 2, method characteristics and how it aligns with the characteristics of the planned study. This can also overlap with the research objectives. 3, target population and availability of the people in that population. You want to consider whether the methods used to elicit information from patients are appropriate for the target population. And this includes the spectrum of the disease or condition, as well as patient diversity. 4, expected short-term and long-term impacts of the information you intend to gather through the study. For example, considering the amount of information that needs to be collected and whether the data needs to be collected cross-sectionally or longitudinally. 5, feasibility of leveraging existing literature and data. Again, this can help with the focus of your research questions and provide evidence to support them. 6, the amount of time that should be used to conduct your studies. And lastly, study budget. This includes staffing, travel time, facilities costs, data storage, management and analysis, etc. Advance, please. Moving on to some specific considerations for some of the research methods. Guidance 2 discusses some best practices for one-on-one interviews and focus groups, as these are 2 common qualitative methods that we generally see used to identify what's most important to patients. So starting with one-on-one interviews, when using one-on-one interviews, it's important to consider patient selection and sample size. This includes selecting sources for participant recruitment for interviews, such as use of medical practices, academic institutions, research consortia, etc. Choosing the appropriate sampling method, which will be discussed in the next presentation, and estimating an adequate number of interviews to conduct. It's also important to consider interview data and collection methods. This involves selecting the appropriate interview type and administration method. This could be in person or remote, and also consider designing suitable interview questions and an interview guide. Pilot testing the interview guide to identify and correct any methodological or logistical issues before prime time in the qualitative study. Interview conduct is another important consideration, which encompasses pilot testing the length of the interview and the appropriateness of the length for the administration method, as well as selecting and training interviewers. So what are the considerations for focus groups? The considerations for focus groups are also similar to those that I just discussed with one-on-one interviews. But there are some other important considerations, which include selecting a trained moderator with appropriate skills, choosing the number of focus groups to conduct, and this may vary based on the complexity and or sensitivity of the discussion topic, the diversity of the participant sample, and a number of planned subgroups. For example, different age groups, disease severity groups, gender, etc. Also, you want to consider determining the sample size for each focus group. Generally, the goal is to keep the group small enough to elicit in-depth responses from each participant, but large enough to get a wide variety of perspectives across different severity levels and demographic representation. So which interview method to choose? So in determining which interview method to use, consider the strengths and limitations of each method, which are discussed in detail in Guidance 2. However, regardless of the interview method, the way the questions are framed is critical to collecting unbiased patient input. At the end of the day, you want to ask the right question. As such, you want to carefully consider the use of prompts, which are open-ended questions to stimulate and provoke participation in a discussion. While prompts can be useful to gain more information from participants, sometimes they can be used incorrectly and come off as leading questions or questions that may imply the desired answer in the phrasing of the question. And this may result in biased or false or misleading answers. And we want to avoid this to the extent possible. Advance, please. Survey research methods are commonly used to collect quantitative data from patients and relevant stakeholders. Important considerations for survey research methods include the choice of survey administration method. It can be self-administration or interviewer administration. And this choice may be driven by a variety of factors, such as study design, target population, etc. Ideally, the survey instrument instructions and questions should be well aligned with the research objectives and designed to answer the research questions, as well as specific to the targeted concept for the research question. Next, consider the format of the survey instrument. For example, formatting it in a simple manner to maximize the ease of use for respondents and interviewers. The assessment of potential response bias is another consideration, as well as pilot testing. This can include both the instrument, but also testing to ensure that electronic or web-based technology is functional and or compatible to administer the survey instrument if applicable. For interviewer administered surveys, consider using a script to ensure standardization. Advance, please. Of course, the considerations for qualitative and quantitative methods we discussed are also applicable for mixed methods. Other considerations for mixed methods should include the sequence of the qualitative and quantitative components, as well as their priority or the dominance. Considering whether qualitative or quantitative methods should be carried out concurrently or sequentially, as well as whether one component should be more predominant in the study or both components given equal priority in the study. Advance, please. So to end this session, we wanted to engage the audience with some questions and also to see if you were awake. So the first question we have here is qualitative research methods, quantitative research methods, or mixed methods research can be used to identify what is important to patients, true or false. And we'll give you a couple minutes to think this over. I'll give you a couple more seconds. And we can share the results. So yes, it seems like the majority of the participants chose the right answer, which is true. Qualitative research methods, quantitative research methods, or mixed methods research can be used to identify what is important to patients. So the next question, next slide, please. Which factors are important to consider when selecting a research method to identify what is important to patients? A, the preference for a research method. B, research objectives and questions. C, target population. And D, both B and C. I'll give a few more seconds. So let's share the results. So again, the majority of participants chose D, which is the correct answer. Both research objectives and questions and target population are some of the factors that you should consider when selecting a research method to identify what is important to patients. So thank you for that. Next slide. So I know this concludes our presentation. And so we want to thank you for your attention, and I'll pass it back to the moderator.
Thank you, Drs. Daniels and Noble. Next, we will hear from Dr. Laura Lee Johnson, who will discuss who to collect information from, sampling plans and strategies. Laura Lee is from the Division of Biometrics 3 in the Office of Biostatistics in the Office of Translational Sciences at FDA. Now I will turn it over to Dr. Johnson. Please turn on your camera and begin your presentation.
Hi, thank you, everybody. And it's nice to see so many people here today. We can go to the next slide. So I'm going to do a brief introduction to sampling. So sampling, we're going to use a few different technical terms, and we're going to talk first about a target population. And the target population is that complete collection of observations we want to study. Now, very rarely do we get to actually talk to everybody. So your target population is something that you need to define when you're going to do the research that was just talked about. And it's true actually for any type of research that you want to do. But let's say, for example, you can take an entire census. You could actually identify everybody in that target. And in some cases, this is true. In some rare diseases, in fact, it is known everybody in the world with that disease is known. And it might be also like for my professional organization, we know everybody who is a member of that professional organization. Although what I may not have a good idea about is everybody who would call themselves a statistician, a biostatistician, a data analyst, etc. But you need to think about what is your target. And if you could collect data on everybody, who is that everybody? Then we want to talk about the sample. So the sample is going to be a subset of this target population. So again, usually our samples are not going to be everybody in the population. And what we're going to talk about in the next several minutes is kind of what are the characteristics of that sample and what type of reflection does that have on what we can do with the data from the sample. Now, there's been a fair amount of back and forth and stretch because people say, you know, I'm not a statistician. I don't do sampling. I'm not going to have a sampling strategy. But just like everything I eat or drink has nutritional value of some sort in it, whether I ignore it or not, every time data is collected, there is a sampling strategy. We just may not think about it. So if we go to the next slide, we'll talk a little bit more about how to think about it to maximize the information from the data that's being collected. So when you read the guidance, we talk about a sampling scheme, and that is selecting that patient population that's going to be participating in the study. And this is really key to get information that's relevant to addressing the research objectives. So if we think about this, if I'm going to do a research study in children, but I only enroll adults, am I getting the information that's relevant to addressing the research objectives? You say, well, if those children were neonates, they've been, their first 28 days of life, I probably do need to talk to those adults. I need to make sure that those adults are around those young children. So we need to think about who are we sampling so we get the right people and the right mix. And there are a lot of different approaches to this. And we actually have some online hypothetical examples on our PFDD website that we developed when we actually had the draft of Guidance 1, where we were trying to help elucidate this balance of what are your study's objectives? What is the objective to collecting this data and resource constraints? Because sometimes you're not going to be able to collect the data that you need in order to address your question. And you should think about that and understand that up front. So next slide, please. There are two really major approaches to how we go about sampling. And I think, again, this is where sometimes things get a little confused, but if we lay it out very simply, it can be very useful. The two major ways of doing this are probability sampling and non-probability sampling. So the probability sampling, there is some version of what is called random sampling. And what that means is that for everybody in that target population, there is a probability attached to that person that they will be chosen to be in your sample. Now, it doesn't have to be the same probability for everybody, and we'll talk about that on the next slide, but that there is some probability attached to every single person. So that means, again, I need to know who I could possibly be selecting. Now, an example of how this works out, I'm going to use some of the CDC large surveys to discuss, but it's also done in some smaller studies as well. Now, you might do something called sample weighting. So sometimes we oversample or we undersample groups. And we may say, listen, I know a subpopulation that I want to make sure I have enough representation from is fairly small. So if I have a sample of 10 people or a sample of 100 or 1,000 people, I need to make sure that I get more than the normal proportion out of that subgroup. And so as you're doing that, you may then, as you're trying to analyze data, use something called sample weights. But this idea of the probability sampling is we are going to sample from the larger population. And because of this, and because we know the probabilities that were used, the results are more likely to reflect the target population. And so from here, what we're going to be hopeful for is that we can make the generalizations to actually talk about the whole target population. Now, what is non-probability sampling? That's a non-random process to select your study sample. And what this means is I don't have that probability attached to every single person in that target population. So that opportunity that they might be selected. Now, the selected sample in a non-probability sampling scheme may not be representative of the target population, but social scientists and many others have spent a large amount of time over the decades to talk about ways that you can try to achieve representativeness. And so we'll go to the next slide and talk a little bit about these. Thank you. So we talk about several different potential sampling approaches in the guidance. I'm going to go through a few of them here. Now, simple random sampling is not commonly done. So I do want to kind of lay that out. It's actually pretty rare, but it is what most people think about when they are talking about random sampling and probability sampling. So here, and part of the problem is that I am going to just simply, usually it's an equal probability that anybody could be selected. And now I'm going to say, like, let's say the first several months. So you may have gotten, if you've been in the hospital recently, you may have gotten a survey from the hospital and they say, please tell us. So they may say everybody who was admitted to the hospital we're going to have a probability that they get the survey. And then you keep trying to find those folks, but it may not be that you actually get the right mix of the heterogeneity of the target population. It could also be that people just don't answer. So we'll talk a little bit about that. The next step that a lot of people use is something called stratified random probability sampling. So here I may say, I want to make sure maybe by disease subtype that I'm getting a random sample. And so this is a way to also help ensure and think about certain individual groups in order to try to do a better job with your sampling, and also to try to ensure that you have a little bit better representation and heterogeneity available. Now, the cluster and multi-stage sampling actually go together a bit. And this is one that, as we're trying to do it mathematically, it requires information about a cluster size. Now, what is a cluster? A cluster might be that hospital in the state. So let's say that I'm trying to find out information on a global scale or on a US scale. I may say this idea that I want to look within states, then look within, and I'm going to try to draw a sample of hospitals within states, and then a sample of individuals who go to a certain hospital. Maybe it's not hospitals, maybe it's schools, maybe it's school districts. There are a lot of different ways that you have a certain type of hierarchy about which you can then cluster and take groups. And so sometimes we're going to take all of the hospitals within, I live in Montgomery County, Maryland, within Montgomery County. So there are different ways of doing selection, but what's also important here is you need to think about the size. In this case, it's going to be the number of the clusters, whether it's a hospital or a school district or a state or a county, but you also need to think about what probabilities I should then be using in order to select that sample of patients inside of it, because you want to, there's different types of math that need to be done depending on if you have different sized clusters or not. Similarly, in the multi-stage modeling is usually that you are trying to go up and down in different types of of layers. And so I mentioned the CDC. The CDC has a lot of major projects that they do to try to assess US public health. And so they may take their information from the US Census. They may take it from Medicare rolls. They may take it because they've accredited those hospitals and they know what the states are. Now, that's a big organization. And you may say, well, how does that apply to me? And the answer is, you actually can apply that in many smaller settings. And so it does depend, again, like if you're trying to do an assessment within your town, within your school district, or across a disease group, what do you know about those patient groups? What do you know about the target population and how people may engage? So now we're going to talk about non-probability sampling. Now here, I will admit, most clinical trials, and I think we had a question about this that came in related in a certain way. Most clinical trials are really a convenient sample, right? So these are people who volunteer to participate. But we have to be careful because it's easy for me to get people at my university in the area where my office is. But that's not necessarily representative of the entire group. So we have to think about it may not be generalizable information. So social scientists many times will use something called purposive sampling. Now here, might be that I'm going to set certain characteristics of that target population. And now I'm going to find people who meet those characteristics. So I may say, I know in general, the demographics are in these ranges. And I'm going to try to make sure I have people that appear to, when I look at the whole sample, have that aspect. So I've tried to make a representative sample based on looking sometimes also at quotas. So we may say your sample needs to have, and it's not usually us, but you may say, I need 45% women in this study. And those are the types of things that these may work together. And sometimes we say a minimum. So, I think we flipped ahead. Can we go back to the last slide? Thank you. So snowball sampling kind of ties in some ways to many of the other types of sampling and non-probability sampling, where we actually take the people that we have gathered a sample, and then we say, who do you know? And this has actually been used fairly well in hemophilia, for example, where they were talking to people in certain settings, and they said, who else do you know? And then they worked out from there and found a lot of patients that were actually very willing and interested in participating in the qualitative research and then quantitative research in order to move forward. So we can go to the next slide now. Thank you. But there are a lot of issues that can come up with any of these methods. Under coverage means that we may, as you've gotten your sample, so plans are great, but then you execute the plan and you look and go, well, I don't have people that fit one of my subpopulations that's really of interest. One of those subgroups, either I don't have enough people or maybe I have nobody. Well, now you have to question, would they have had different answers? Can you generalize or not? And if you don't have information, it's hard to tell that. Same thing with non-response and dropouts. So people may be selected, they may decide they don't want to participate, or they may make a decision that they start listening to your questions and they leave the study, or they may not answer all of your questions. And then especially when you're talking about your non-probability sampling, there's something called voluntary response bias. So remember, the people who agree to be in a study may not be at all like the people who are in the larger group. So if we can go to the next slide. All of this ties into our ability to make generalizations from the data. Nothing is going to be perfect, but can we take some steps that will allow us to improve the generalizability and representation? And this all ties into our sampling schemes. Next slide, please. So can we generalize to the target population? This is the key question when we are trying to review data that we ask ourselves. Are the subgroups adequately represented in the study sample? The various characteristics that do they approximate the heterogeneity and the characteristics of the target population? These are questions that we are trying to evaluate when we are doing our reviews. And that's true for any type of patient experience data and for other types of data too. And is it feasible if it's not matching up? That weighting could be used to account for either over or under sampling, in particular, if that probability sampling was used. So if we're going to do weighting, we need to understand the probabilities of the selection or inclusion of people from that target population into the sample. We also have to think about non-response. And so it's common that folks are thinking about how to handle that type of data, but also thinking about, at the end of the day, what are the differences in that final samples population and the target population? Because while you may have great plans, you also need to think about what happens at the end. And so a lot of investigators will look, and they will be looking piece by piece, step by step, to say, do we need to do more targeted recruitment? Do we need to target in other ways? And so now let's go to the next slide. And again, part of what we're trying to consider here is that is this sample now representative of the target population, and how far off is it? And do we think it's going to matter? Patients in the study sample should reflect the diversity and the heterogeneity of the patient characteristics in the target population. However, as I mentioned, that distribution of the characteristics in the sample, it could be different than that of the actual population. So it's not that there's a failure if the characteristics are different. It does, however, then make you think like, okay, what do I need to consider as I'm doing my analysis, as I'm trying to make decisions? Because sometimes, in fact, we need information. There should be a range. Very rarely do we expect to hear everybody say the exact same thing. It does occasionally happen. I had an investigator who came to me and said, he's like, everybody's describing it using the same word, and it doesn't matter. And he gave through all these examples, and he was right. That was the data. And that was good to know, because now we had a path forward. But usually you're going to have a range. And you need to consider now, based on the objectives and how that data is going to be used, if you're ready to move forward. And this is another part to consider, which is depending on where your data may be used, what, again, are the objectives of the study? Who are the decision makers who are going to be looking at the information? But also think about not just your objective right now, but where else might this data be used? We are in a time where, and it's in many ways good, patients shouldn't have to participate in caregivers as well. In 20,000 little studies, can the data be reused? But that also means thinking about the purpose and where you're going to go. What are the objectives? How are we going to handle this work? So if we go to the next slide, we also have to think about missing data. So when people are not responding, they don't want to participate in your survey. I know I've done it before. Or it might be that you have other things that are happening, but this impacts the representativeness. If they decline to participate, if they drop out, if they've stopped participating in your study, if they decline to answer some of the questions, this is going to impact your work. So what you should do up front is anticipate what is likely to occur and also what barriers can be removed. And this is a great place to include the very people that you are going to be talking to. Why? Because they're going to tell you a lot of information that might be uncomfortable, but is necessary. The best information I have learned has been from patients, it's been from the nurse coordinators and the people at the front desk, because they're going to give you the straight story about what's happening. Study design features and those, like what's going to work and trying to talk to people. Is it going to be too long? Is it short, too short? Also logistics, the time of day, the location, how they can interact, who they are interacting with. There are a lot of logistics that make participation difficult, and you may not be thinking about them or recognizing them. But in fact, sharing those learnings are some of the most important parts of sharing that we can do in trying to move the patient experience data field forward. Also, the specific data being collected. There may be some things that you do need to ask about, even though they could be kind of taboo subjects. So part of my work is doing a lot of work with bowel movements. Also, we do a lot of work where we have to talk about sex. There are cultures and places that they do not want to answer those questions. But if we're going to need that information in a clinical trial, part of what we then have to figure out is how can we gather the information? How can we approach it? How can we explain it? What can we do to remove barriers but still achieve the objective, or to tweak our objectives to make sure that we're still going to get to a place that is going to be okay. Another part that's gained more momentum and that we strongly encourage is, as you are going through your research, is determining the reasons for missingness. Why did someone stop participating? And telling them up front, like, we're going to ask this question because it's going to help us do a better job in the future. Figuring out, like, are there certain questions that people are declining at different rates? Can we figure out why? It's a constant learning process, not just about your main research objectives, but about doing the research itself and working with and in your target populations. And also understanding then the extent and the impact of any missing data, non-response, et cetera. It might be that in fact, you don't have an impact, but it's not just numbers alone. If you're missing key people, that may be a very small part of your sample, but have a huge impact on the representativeness of what you are doing. So we go to the next slide, please. May we go to the next slide, please? So just in case you're seeing it, but I'm not, our next slide is on the intersection of representativeness and diversity in sampling. So I think it's important for us to consider what are the attributes of interest, the socioeconomic and demographic backgrounds that we're interested in, and thinking about the cultural backgrounds, the spoken languages. So when we're trying to think about what is, thank you. When we're trying to think about what is the type of questions when we are asking questions internally, I wanted to bring these up. So people talk a lot about diversity, they talk about inclusion, they talk about equity, they talk about being able to actually engage. If you are in a situation where, for example, people are using assistive devices, do you have the variety of assistive devices and people that are using them in your sample? What are the attributes that are of interest? It's not always going to be gender, age, race, ethnicity. It might be, but you're going to probably have to go past that, because samples that are just fine, like, will look at me like, oh, pretty much everybody in your sample has a master's degree. That's unlikely to be a very representative sample, even if it is a diverse one. So you need to think about also, where is the study going to be done? If you're only going to do interviews in English in the US, is this going to then inform really well a PRO that's going to be used in international trials? Again, thinking about what are the attributes that are of interest to you? What is the long game of where this data is going to be used? Also thinking about the literacy and health literacy, and I didn't write down numeracy but it is an important part, especially as we look at some of the preference trials and other trials that are being set up. If you're trying to do an assessment, are people really understanding the question? But that's on you. It's not on them. And I think it's very important for us to think about, how are we getting the information, and are we actually getting the information we think that we are getting? But also thinking about the clinical characteristics. Those clinical characteristics are really important because, again, if you've only done your work in people that have really severe disease, and now you're trying to actually apply it in people that have mild or moderate disease, they may be very different. So think about who it is you are including, but also being very honest about what's being excluded. Next slide, please. So we're going to leverage our existing data. This is, again, as I mentioned earlier, it's a big thing, and we encourage it. But we also have to demonstrate, I should say, and not but. When it comes in, you've got to demonstrate the representativeness of that data. So you're coming in to say, hey, this is our registry. This is the registry, it's what we have. But the registry doesn't have a representative sample. Well, what does it represent and what is not there? We need to understand those demographics. And again, beyond to all of those attributes that are going to be really important and of interest. The methodological rigor of the data collection methods, and also the data integrity, the data quality. What is there? How did it get there? Not only do we at FDA need to know it, and EMA and other regulators are interested too, you need to know it if you're going to be using it. And if you're collecting this and you want people to use this data, you need to have those documents available and ready, and also think about, how can I improve my data set? Because I do think it's important that we leverage this existing data, and at the same time that we're trying to elevate our data collection methods across the board. So if you are part of the group that collects and creates and curates that information, these guidances are very important for you, because it really tries to lay out how we can best have and expect that data to be coming to us. So on the next slide, I'll start into my summaries. We have objectives at the beginning, we have a purpose at the beginning, and we make plans tied to that. But at the end, we have what we have. And we don't know what we don't know, and what we don't hear, and what we don't see. So if we go to the next slide, our main considerations, if I'm going to boil this down, is to focus on the purpose and objectives, also thinking not just right now, but where all of this data collection can go in the future. Who do we need to have involved? But also keeping in mind, what does the decision maker need and what is going to be useful for them? And budget in time to talk to the decision makers. And they could be a variety of people. It might be, leadership inside of a company. It might be inside your patient group. It could be regulators. It could be payers or other health organizations. But budget in time to try to see if where you think your data is going to match, it's going to actually match. And we have a little bit about that in those online examples. Next slide, please. I want to conclude my section here in saying that all of this is important, and it goes back to one of the very first things I said, because participants' time matters. And we must be using the methods that can answer the questions that decision makers are trying to answer. And I'm not saying only regulators, any decision maker. We need to make sure that the research that's moving forward actually can address those questions and the purposes that it's there for. Because if the research is not going to actually answer the question, it's probably not worth the time, the money, and the energy that could be spent answering a question. So really do focus on the purpose, the principles, and the science, because everybody participating in that research their time, their energy, their money also matters. It takes me time out of my workday to participate in a clinical trial. That's time, but it's also money for me. It's money for you. It's for everybody. So really stay very focused with good science and good principles, but make sure that they are addressing the purpose that you have. And with that, Shannon, I'm going to turn this back over to you.
Thank you, Dr. Johnson. Next, we will move on to the second session of the meeting, where we will hear from external stakeholders on these ideas put into practice. First, we will hear from Dr. Ebony Dashil-Ajay from BioMarin. Now I will turn it over to her. Please turn on your camera and begin your presentation.
Thank you so much, Shannon. Good afternoon, everyone. It's my pleasure to just talk to you all about some industry applications of Guidance 1 and Guidance 2, particularly as tools to generate patient experience data to support medical product development. Next slide, please. First, I'd like to applaud the agency for endeavoring to provide these methodological guidances to the public to help facilitate the generation of high quality data and also to facilitate the bidirectional communication between the agency and sponsors during the product lifecycle. Overall, Guidance 1 and Guidance 2 have been helpful for driving development of robust, meaningful, and interpretable data on patient experiences, perspectives, needs, and priorities to help support medical product development. And from an industry perspective, we found these two documents most useful for guiding study planning, preparation, and methodological implementation. And we've gleaned many takeaways, including, for instance, that we need to establish clear research questions and objectives at the onset. We need to ensure representativeness and sampling to generate insights from the appropriate target population. We need to also select the right methodology to generate the right data in the right patient population. We also need to determine which method, whether qualitative, quantitative, or mixed methods, should be used to understand what is important to patients. And lastly, we know to establish best practices and follow them in order to generate reliable and valid data in every study that we conduct. Next slide. Now I'm going to go into some high-level principles from Guidance 1 and Guidance 2, starting with the guiding principle of making sure that we are obtaining reliable and valid patient experience data in the right target population. So who are we asking these questions and how are we sampling and ensuring representativeness in our studies? At BioMarin, this involves reflecting on the population under study in our clinical trials and designing complementary studies with the same population in mind, with the goal of generating insights among the broadest range of the target population as possible. And in rare disease drug development in particular, it's important to carefully consider how the small sample sizes may pose challenges to recruitment, especially in geographically dispersed and heterogeneous populations. And in considering the guidance recommendations, we've considered multiple demographic, cultural, literacy, and clinical characteristics to define our sampling strategy when we're designing these complementary studies. And we then use a multi-pronged approach to recruitment to ensure that we're able to include the most appropriate subjects in our work. And this means that we partner with external stakeholders such as patient advocacy groups, as well as investigative sites to make sure that we develop rapport with the community and we also are sampling the most broad range of patients that we can. We also have used other patient-centric for-profit organizations like Savvy Cooperative and the like, as well as social media strategies to bolster our recruitment efforts and increase our reach. Next slide, please. Based on Guidance 2 principles, we've also taken a very pragmatic approach to identifying research questions cross-functionally in our organization to determine the exact type of data that would be most appropriate to support our program goals and apply those best practices to generate data that is suitable for regulatory submission. As my colleagues so eloquently mentioned and outlined, FDA has been very open to the generation of different types of data, including quantitative data through like surveys and questionnaires, as well as qualitative data generated through interviews and focus groups. And even the guidance mentions methods like ethnography that could be useful for generating deep insights about the patient experience. Likewise, mixed methods are encouraged in the guidance, and we regularly use integrated methods to qualitatively generate data, to qualitatively interpret quantitative data, and be able to use this to guide our discussions with regulators on our study design and study goals. Next slide, please. While not explicitly outlined in very much detail in the guidance, we know that the guidance has outlined frequent and early communication with the agency about our study plans in order to fully align on the best objectives and goals to meet regulatory needs. And as a result of the guidances, trade organization partnerships have been integral in helping us to outline exactly when to generate the data that guidance 1 and 2 have outlined as important. As you can see with this particular chart that has been generated through BIO, there's a very comprehensive mapping of the types of methods and the types of data that can be generated at different milestones throughout the medical product life cycle. And these types of roadmaps have been particularly useful to help us digest and interpret the contents of these guidances. And one thing I want to highlight too is that even FDA has heard the cries of industry and has carved out another type of meeting, a type D meeting that could be used to discuss patient experience data going forward. That's not currently included in this roadmap, but it's worth highlighting, of course. Next slide. Now I'd like to just go into some specific case studies on where we've applied the guidance principles to generate very much reliable and valid data that has helped us move forward with our drug development programs. The first one is when we've been trying to establish unmet need and measure and identify measurement gaps in HAE. So hereditary angioedema is a rare genetic disorder characterized by unpredictable, painful, recurring, and self-limiting acute edema attacks that may occur at multiple locations on the body, with the most debilitating and life-threatening of attacks being laryngeal attacks. And while the use of prophylactic therapy has been shown to substantially reduce the number of attacks patients experience, patients may still experience severe breakthrough attacks that can cause debilitating pain or significant morbidity and can be life-threatening to them. And additionally, available treatments may be effective in reducing the time to improvement of symptoms. However, patients still remain symptomatic until attacks are resolved. So there is a remaining unmet need in this patient population. And as we've worked to design our gene therapy study, we had to use the guidance to help us understand how to generate the most appropriate patient experience data, to characterize unmet need, and to design our trial in the most robust way so that it will be acceptable to regulators. And in that process, we used guidance 1 in particular to help guide how we would determine the most appropriate target population for our trials, as well as for the complementary studies that we were using to generate data that would help move the study design forward. We engaged the patient community to understand their perspective on unmet need, and then taking all of that information, we were able to develop a data and evidence generation plan focused on specific research objectives that were aligned cross-functionally. We established the need for targeted lit review, gap analysis, advisory boards, and qualitative interviews to gather relevant patient insights. And ultimately, we used that data to conduct our relevant studies, and then ultimately to present to the agency to support regulatory discussions. Next slide. Next slide, please. Thank you. The next case study I'll highlight is in PKU. Now, PKU is a rare metabolic genetic disorder that can cause various neuropsychological symptoms that often affect patients' health-related quality of life, even for patients with good metabolic control management. And, it requires lifelong adherence to Phe-restricted diet or metabolic food intake in order to keep Phe levels at a near normal range. And we know that treatment adherence in this patient population is often challenging, but also it's been very difficult in order for us to measure the appropriate neuropsychological impacts for this specific patient population. And as we've gone to develop medicines in this area, we identified that there was still a gap in terms of how best to measure the most relevant concepts in this patient population and how to apply that within clinical trials. So in order to determine how best to establish clinical benefit in PKU, we embarked on developing a de novo tool that would be very specific, a disease-specific tool to measure the direct neuropsychological symptoms and quality of life impacts that were specific to this patient population. So in order to develop this tool, we engaged the patient community to understand their perspectives on unmet need. We identified existing measurement gaps, particularly that there was no PRO that exists to combine both the symptoms and the impacts as well as aspects of diet control into one measure. We conducted qualitative interviews among clinicians and patients. We used information generated also from an advisory board to inform conceptual model, et cetera. And ultimately, we developed this draft instrument, and now we're embarking on validation work so that we can put it into the field. Next slide, please. Another example of guidance application was a stepwise development of a patient preference survey to gather perspectives on disease and treatment experiences, as well as personal preferences and relative importance of treatment characteristics to drive and inform treatment choice among patients with hemophilia A. Now, Guidance 1 and Guidance 2 are not dedicated to the discussion of specific patient preference methodological approaches. However, in the broadest sense, patient preference studies, especially discrete choice experiments, involve stepwise mixed methods approaches, including both qualitative and quantitative data collection. And as such, the foundational work required to assess patient preferences require the application of principles outlined in both Guidances 1 and 2. And here you see a screenshot of a poster highlighting results from the first qualitative phase of our discrete choice experiment study conducted in patients with hemophilia A. And for this portion of the study, we conducted semi-structured interviews, concept elicitation telephone interviews as well, and gathered insights on ideal treatment outcomes and perspectives on potential risks of treatments. We also generated ratings and rankings of predetermined treatment attributes from not important to very important or most important to least important to truly determine what trade-offs and what things patients are weighing when they're making treatment decisions. Next slide, please. And lastly, I wanted to cover other ways that we can elevate the patient voice in a complementary way outside of the very systematic qualitative or quantitative methodologies. As outlined in the guidance, the patient voice and patient experience data is very broad. Therefore, there are a number of things that the agency has articulated that they consider when trying to make drug approval decisions. And from BioMarin's perspective, we know that you can gather information qualitatively from the patient community through the traditional methods of interviews and focus groups and ethnography. But sometimes all you need to do is just ask them, and they can summarize in a very fluid way what their experiences are. So we've partnered with external stakeholders, patients, caregivers, advocacy leaders, clinicians, KOLs, to help them have a platform to tell their stories via oral or written statements, videos, different presentations. We've empowered the community through knowledge about how best to interact with the agency independently, but also we incorporate their voice into regulatory milestone meetings and submissions. We also partner with these stakeholders in COA development to amplify the patient voice. And the guidances have been very clear about being able to generate all kinds of patient experience data. And we appreciate these other methods being highlighted and this data being used in regulatory decision-making. Next slide. Although FDA has made great progress in providing guidance to stakeholders on what constitutes sufficient evidence to reflect the patient voice and to support regulatory decision-making, we can still benefit from more progress in the areas that I'm outlining on this slide. For instance, there's room for greater transparency on what type of patient experience data is considered acceptable to support regulatory decision-making, especially when applying regulatory flexibility in the rare disease drug development. Likewise, we could benefit from greater guidance on how more novel patient experience data like patient preference information or testimonials, ethnography data, video documentation can support regulatory review throughout the medical product life cycle. We also would benefit from understanding like the best timing for discussions with FDA on these types of data. Lastly, we think that there could be some progress in development and sharing of pragmatic approaches through publicly available examples that can help stakeholders tackle common challenges to applying regulatory guidance on patient experience data generation. And with that, next slide. I'd like to acknowledge all of the work that my colleagues at BioMarin have been involved in to generate these case studies that I presented today. Thank you. Now back to the moderator.
Thank you, Dr. DeSheila J. Next, we will hear from Dr. Becky Noel from Eli Lilly and Company. Now I will turn it over to her. Please turn your camera on and begin your presentation.
Good afternoon, everyone. I wanted to give an update on the progress of science, on the science of patient input. And we will be looking at the guidance documents 1 and 2, as well as a European effort known as IMI Prefer. Next slide, please. So thanks to all of the presentations that have come before me, I think we've heard mention of some of these efforts in these other presentations. But really what we wanted to do in this slide and the next is outline the evolution of the science of patient input. Many of you may be familiar with the efforts in the US and in Europe to develop a systematic and structured approach to benefit risk assessment. That effort goes back now well over 15 years. We saw as an output of it, the FDA develop a benefit risk framework, which they now use to guide their reviews of new drug applications. And in parallel with that effort to develop structured and systematic approaches to benefit risk assessment, there was a parallel effort to simultaneously begin collecting patient input in a way that could better inform drug development and review decisions. Now on this slide, you'll see an alphabet soup of activities, but really what this slide is intended to show is the broad range of efforts across multiple organizations and the FDA and the EMA. Next slide, please. Similarly to the efforts that we saw from the FDA and from the EMA, we saw efforts from a wide variety of patient organizations, patient communities, and public-private partnerships to strengthen the degree of patient experience data that can be used to inform drug development, drug review, and even reimbursement decisions. Next slide, please. But as you saw mentioned throughout the presentations today, there really was a common thread that tied together the industry efforts, the regulatory efforts, and the patient community efforts. And that really was this explicit recognition that patients who are living with a disease are uniquely positioned to relay their perspectives and preferences and thus contribute to a drug's development and its availability. Next slide, please. So that brings us to the primary reason that we're here today, and that is a review and reflection on Guidances 1 and Guidances 2. I think many are aware that Guidance 1 was released in 2020 with the final guidance for methods on released or finalized in February of this year. And really, I think it's important to reflect on the purpose of these, and that is that Guidance 1 was about methods to collect accurate and representative patient experience data, with Guidance 2 focused on helping us be able to identify, characterize in an evidentiary way what's important to patients with respect to their burden of disease, their burden of treatment, and then the benefits and risks. Next slide, please. But if we want to take an even higher view of the efforts around developing the science of patient input, we know that FDA has not been pursuing these efforts alone or in isolation, that really there is a transnational effort by many regulatory authorities to develop the science of patient input by strengthening benefit risk assessment and really strengthening the development and evaluation of patient experience data. Ebony mentioned a specific subtype of patient experience data, which we refer to as patient preference information. And really the case study for patient preference information that we'll take a little detour into recently came out of the Innovative Medicines Initiative PREFER effort. And really PREFER was a nearly a 7-year-long effort that looked at how and when to perform and include patient preferences in decision-making. And that decision-making included development, regulatory review, and reimbursement. Next slide, please. So taking a bit of a side tour into a specific type of patient experience data, what is patient preference information? Well, patient preference information is information that captures the value that patients place on various aspects of medical treatment. Patient preference information can account for differing patient perspectives on the benefits and risks, which we refer to as attributes of treatment, that come with using that medical product. And Ebony was kind enough to point out that PFDD Guidance 2 specifically states it doesn't address methods for collecting and analyzing PPI, but it does discuss best practices in performing qualitative research. And if you look at this information around best practices, you will absolutely see that the core commonalities that identify and describe and help us understand what best practices look like certainly apply to patient preference information. Next slide, please. So as a component of IMI Prefer, we chose to pursue something which is referred to as a qualification opinion. And the reason that we did this is because the usual output of an IMI project is a set of recommendations. And IMI Prefer certainly did produce a set of recommendations. They're very robust and they provide a wealth of information for anyone thinking about or wanting to learn more information on patient preferences. But we wanted to do something more than deliver a set of recommendations. We wanted to be able to provide a framework and points to consider on a selected set of methods that those who were looking to develop and include a patient preference into their new drug application, into their submission, could use as a roadmap for how to do that. And we needed something that took the recommendations and actually actioned them for industry, for patients, and for other stakeholders. And so we pursued what's known as a qualification opinion. And a qualification opinion means that you submit a tool or a method or a process to the regulators, in this case, the EMA, and the regulators evaluate that for a base level of suitability for use in a submission. And so partway through the IMI project, halfway through the project, we began the work needed to submit the Prefer framework. The Prefer framework lays out a set of principles and tools for the development and use of patient preference information. Next slide, please. So in the qualification that we submitted, two of the things that we highlighted certainly were method selection and analysis, as well as a detailed and comprehensive discussion on the use of qualitative research methods. And we wanted to do this because we know that qualitative certainly informs the quantitative, and that, as Ebony highlighted, preference work is a nice example of mixed methods. Next slide, please. We also submitted, as part of the methods, the framework, and the framework outlines also points to consider for the development of both the qualitative aspects and the quantitative aspects of any patient survey. Next slide, please.
Thank you, Dr. Noel. I'm sorry to interrupt, but I know we are approaching the time allotted, and
yes,
and we're
absolutely almost there. So to those of you who are looking to learn more, that you can find at the end of this slide directions that will point you not only to the framework but also the qualification opinion. We can move through this slide. I think Ebony addressed that, but really we wanted to point out as part of understanding the importance of the development of qualitative methods, that you see that reflected not only in the FDA, but also in the EMA work. This is just our woohoo slide, in which we can point out that in May of this year, the EMA did indeed issue a positive opinion, and the IMI prefer framework endpoints to consider for methods have been endorsed and are there for industry and stakeholders to use in their submissions. And next slide, please. I think that might be it. So just as the final slide, we want to point out that there are very significant corollaries between the work that we see happening not only at the FDA, but also at the EMA. And as was mentioned, perhaps by Teresa at the beginning of the presentation, this work certainly will not stop here. And we're looking forward to supporting this and progressing it, hopefully to ICH. And with that, I'll turn it back over to the moderator.
Thank you, Dr. Noel. And I'd like to say that I know we are running a bit behind schedule, and that will impact the length of our Q&A session at the end, but we are working behind the scenes to answer questions in that Q&A chat through Zoom. So you should be able to see those answers as we post them. But moving forward, next we have Dr. Belinda King-Kalamanis from Longevity. She will share her perspective from the experiences of a patient organization. So I will turn it over to her now. Please turn on your camera and begin your presentation.
Thank you, Shannon. And I'll try and make up some time here to get us a little bit more on track. So I'm going to be speaking from the patient advocacy perspective on guidances 1 and 2 today, which are great resources for everybody. Next slide, please. So my fellow panelists have dove deeper into the guidances, but now I'm going to take us back to thinking about representation and what it means for instrument development. So when we're developing items for patient-reported outcome measures for drug development, we always talk with patients who are living with the disease. However, ensuring our samples are truly representative of all patients who might end up in a clinical trial is a real challenge because good research takes time. And the reality is that there isn't a lot of time allotted for the development of especially a new PRO instrument, but even if we're testing one out in a different population. But in a study that looked to speak to trial coordinators to understand some of the facilitators and barriers to PRO completion as part of a clinical trial, the researchers heard things like, I think sometimes the questionnaires are designed by people who don't have a lot of patient contact. Sometimes you need to highlight things like, for example, we want you to complete this as how you've been feeling in the last 7 days. And it's true, as researchers, we don't talk to patients day in and day out. Or I usually say question number such and such has been missed. You haven't given a response. Is that because you weren't sure of how to answer or because you didn't want to answer that question? And they go, oh geez, I didn't see that one. Or nausea, what does that mean? Next slide, please. And if you're thinking that nausea might not turn up so much these days, you'd be wrong in that here it is in the Pro-CTCAE measure. And if you were listening yesterday to the Oncology Center of Excellence's workshop on clinical outcome assessments in cancer clinical trials, you would have heard a lot of talk about this instrument. And yet here it is asking in the last 7 days, how often did you have nausea? And I'm not saying whether it's a good or bad instrument, it's more complicated than that, but it does still turn up. Next slide, please. So in guidance 2, it says that we should be thinking about qualitative research methods, quantitative research methods, and mixed methods research to identify what is important to patients. And so our samples really should look more or less like those with the disease or using the treatments that we're trying to understand, that is that target population. But in reality, a lot of the time, we need to develop something quickly, and it means our samples often include more of the first to sign up. In other words, our samples lean towards being convenience samples. And as Laura Lee talked about, you really do have to be aware that who you're sampling from and where they come from in the larger population of those living with that disease. And a convenience sample is not likely to be representative. Next slide, please. So in Guidance 1, there's this great figure, and Laura Lee again did touch on this too, that gives the kind of things we should be thinking about to achieve sufficient representation. And this is quite a list to try and get to, especially on the timelines we're thinking about with sometimes very small sample sizes. So thinking about those socioeconomic and demographic backgrounds, and that there's intersectionality here as well that we need to consider, as well as cultural backgrounds and spoken languages, literacy and health literacy. This is a real challenge to screen for. We don't have a lot of great instruments. There are some, but I know in our own studies at Longevity, we've not been sure how to really tackle this. And then clinical characteristics, ensuring that you have a range of severity of disease or the condition you're thinking about, the symptoms that their patients are experiencing, comorbidities, and their physical and cognitive abilities as well. Next slide, please. And so if you come to me at Longevity and are looking for some extra patients to fill in your samples, what I can get you really quickly is this list here. And this does depend on the disease and context that you're thinking about, but we don't really want to have our samples filled with people from higher socioeconomic backgrounds and graduate educations, regardless of the disease area. And that's how words like nausea get into our questionnaires and surveys. But in lung cancer, most patients are older, if we think about the entire population living with lung cancer. But what I can find you are those who are younger and healthier, which does look like the clinical trial sample, but we are trying to expand that eligibility criteria and include a more representative sample in our clinical trials. And they'll live in cities, be predominantly female, and whites are very much so overrepresented. Next slide, please. So the way I see this is that we have a little bit of a problem. We use a one-size-fits-all approach in our outreach to patients to participate in our PFDD studies. So when we invite patients, it's not always clear to them why their particular voice is important and what it is that they're really bringing to the table. And I think this is because we don't always involve patients in the development of our outreach materials. And sometimes our outreach materials are even a secondary thought. The protocol, the statistical analysis plan, or the discussion guide take more precedence, but really we do need to be cognizant of how we are talking to patients about our studies. And while IRB requires non-coercive language, that doesn't say we have to use the same language for everyone. Next slide, please. So if we think about how public health campaigns are run and how they try to reach a broad swath of people to either inform or change behaviors, there are researchers involved, but in addition, there's usually market research folks and advertising people who have learnt from their work that, generally speaking, perhaps women respond to emotions more, whereas men will respond to functionality and reputation. And in this case study that looked at smoking cessation behaviors for seeking out information, they found that men were more likely to conduct smoking cessation searches when exposed to advertisements containing empowering content, whereas women were more influenced by ads emphasizing health effects of smoking. So how we influence health behaviors differs for different groups. Now, I'm not saying we should develop 80 different messages, but at the moment, we're really relying on one message. So we can certainly have a little bit of room to expand there. And so we should be involving our patients in developing our outreach materials. Next slide, please. And so to conclude, it says in Guidance 1, you should examine previously conducted studies and other relevant research literature and consult subject matter experts, e.g. clinicians, social scientists, patients, advocates, caregivers, to help determine the most appropriate question. And I would add that when you include patients on your advisory group for your research, also include them in reviewing the outreach materials to ensure potential participants understand the value that they bring to this really important work. And with that, I will wrap it up. Thank you.
Thank you, Dr. King Kalamanis. Now we are going to switch gears a bit and hear from FDA staff member Dr. Erica Lyons on a clinical regulatory perspective on guidances 1 and 2. Dr. Lyons is a clinical reviewer in the Division of Gastroenterology in the Office of New Drugs at FDA. Now I will turn it over to her. Please turn on your camera and begin your presentation.
Hi, good morning. Thank you so much. And thank you for inviting me to participate in this meeting. As you said, my name is Erica Lyons. I'm actually an Associate Director for Therapeutic Review within the Division of Gastroenterology. And today I will be presenting a clinical regulatory perspective on the implementation and impact of patient-focused drug development. Next slide, please. And next. Before we begin, I have no conflicts of interest and nothing to disclose. I do have to state that this talk in some cases reflects my own views and should not be construed unless noted to represent FDA's official views or policies. Additionally, for the ease of communication, I will be using the term drug to refer to both drugs and biologic therapies during this presentation. Next, please. So again, on behalf of my entire division, thank you for inviting us to participate in this meeting. We are all aware that assessment of the patient voice has become recognized as a vital component in the evaluation of new drugs. And today I'm very pleased to be able to share with you how the indications that we manage in the Division of Gastroenterology have directly benefited from the implementation of elements contained in PFDD Guidances 1 and 2. Next. As required by the 21st Century Cures Act, FDA conducts regular assessments of the use of patient experience data in regulatory decision-making. Now, on June 18th of last year, Eastern Research Group, as contracted by the FDA, published an initial report on the FDA assessment of the use of patient experience data in regulatory decision-making. Now, this assessment included data from 1,169 new drug applications, or NDAs, biologics license applications or BLAs, and efficacy supplements, and covered applications received by FDA between June of 2017 and June of 2020 that were approved by February 5th of 2021. Of these, 176 applications were submitted for novel drugs that had never been previously approved. And these are commonly referred to as new molecular entities or NMEs. 68% of the FDA reviews for these NME products describe patient experience data either from the application or from other sources. Next. There are many types, as you heard this morning earlier, of patient experience data. And this table shows the types of data that were mentioned in the reviews described in the 2021 report. And you can see that of these applications where the type of data was identified, patient-reported outcomes, or PROs, that measure how patients feel or function through the assessment of clinical signs and symptoms based on a report that comes directly from the patient, were by far the most common of the assessments described. PROs and other clinical outcome assessments, or COAs, are also the most likely to be considered in benefit risk analyses and used as endpoints in clinical trials to support approval decisions, while other types of patient experience data are more often used to understand patient priorities and preferences, the disease or condition, or burden on patients. Of the applications for gastrointestinal disorders represented in the data shown, 80% had patient experience data considered in FDA reviews. Next. Let's move on to why PROs are so commonly used in clinical trials for GI disorders. Clinical benefit is a positive clinical meaningful effect of an intervention. That is a positive effect on how an individual feels, functions, or survives. FDA-approved drugs have shown both benefit or efficacy and safety for patients with the disorder that they're approved for through studies that were appropriately designed to distinguish the effect of a drug from other influences or factors. In many GI conditions, more objective outcome measures such as irreversible morbidity or death occur infrequently. As such, they are often not practical or feasible to assess as endpoints for clinical trials. And this is due to the extensive time and resources needed to conduct these trials adequately, including the large number of patients that would need to be enrolled and followed for many years to see a treatment effect. Alternatively, these patients commonly experience clinical symptoms as primary manifestations of their disorders, including symptoms such as constipation or diarrhea, heartburn, and abdominal pain. And these symptoms often have substantial impact on the patient's abilities to function on a day-to-day basis. As a result, clinical trials for GI disorders frequently utilize alternative outcome measures, such as PRO assessments, as primary or co-primary endpoints to demonstrate that a drug leads to clinical benefit. Next. With this context, it's not surprising that recommendations to position PRO assessments as primary or co-primary endpoints for GI drug development programs have been recently outlined in FDA guidance for several GI disorders, including that for eosinophilic esophagitis, celiac disease, and the symptomatic treatment of internal or external hemorrhoids. Recommendations for the development and implementation of PRO assessments are also discussed in the recent draft guidances for ulcerative colitis and Crohn's disease, both of which were published in April of this year. These guidances were published to describe the agency's current thinking and assist sponsors as they pursue the development of drugs for the conditions by providing a recommended approach. Next. From these examples, I'd like to expand on the implementation and impact of PFDD for the development programs for the treatment of eosinophilic esophagitis, or EOE. EOE is a chronic allergic or immune-mediated disease that's part of a larger group of disorders known as eosinophilic gastrointestinal disorders. In patients with EOE, large numbers of white blood cells called eosinophils are found in the inner lining of the esophagus when this tissue is looked at under a microscope. Normally, there are no eosinophils in the esophagus, and this eosinophilic inflammation is referred to as histologic activity. Patients with EOE also have clinical symptoms, and these vary with the age of the patient. Infants and toddlers present with feeding difficulties, school-aged children are likely to have vomiting or pain, and adolescents and adults report dysphagia or difficulty or pain with swallowing and risk of food impaction or food getting stuck in the esophagus. Untreated, EOE leads to esophageal stricture, which is an abnormal narrowing or tightening of the esophagus. And by current estimates, EOE affects somewhere between 1 to 2 per every 2,000 people. And that roughly translates from anywhere to 166 to 332,000 people in the US today. In patients with EOE, clinical symptoms or features and histologic activity can vary independently. And for this reason, treatment goals of EOE include resolution or reduction of the signs and symptoms of active disease to provide relief to the patient, and healing or control of the esophageal inflammation and its complications. Next. In September of 2020, FDA finalized the guidance for industry on eosinophilic esophagitis, developing drugs for treatment. A key component of this guidance is the recommendation to assess co-primary endpoints to support the efficacy of a drug for the treatment of patients with EOE. These endpoints should include significant and clinically meaningful improvements from baseline in signs and symptoms as compared to placebo, using a well-defined and reliable COA instrument, and documentation of histologic response as defined as a peak eosinophil count of less than or equal to 6 across all available esophageal levels. As a co-primary endpoint, inclusion of a patient-focused COA in these clinical trials is considered essential to demonstrate clinical benefit and support both potential drug approvals and labeling claims. Additionally, in the guidance, FDA encouraged COA development to begin early in the drug development and provided recommendations for anchor-based analyses to aid in the interpretation of the COA endpoint results and whether an effect was clinically meaningful. Next. Now, although several COA instruments have been developed to evaluate signs and symptoms of EOE in clinical practice, there are no publicly available assessments that have been shown to be fit for purpose for regulatory use. As such, the recommendations detailed in PFDD Guidance 1 and 2, shown here and discussed earlier during the meeting, are critical to support the collection of robust, meaningful, and representative patient input to inform both the development of PRO assessments in trials of patients with EOE, as well as the anchor assessments to be used in these trials to enable the interpretability of the clinical meaningfulness of any treatment differences observed on assessment of the PRO. Next. Now for the exciting part of my presentation. On May 20th of 2022, dupilumab became the first FDA-approved therapy for the treatment of adult and pediatric patients ages 12 years and older with EOE. I'd like to highlight the following excerpts from the FDA news release that discuss the symptomatic co-primary endpoint that supported this approval. The development program for dupilumab used a PRO assessment called the Dysphagia Symptom Questionnaire, or DSQ, to measure how patients felt and functioned through the assessment of the clinical symptoms of dysphagia based on a report that came directly from the patients. The DSQ was developed and was selected for use in this program based on qualitative and quantitative data that identified and supported dysphagia as a concept and symptom that was representative of the patient experience and important to adults and adolescent patients with EOE. The results from Part A and B of the pivotal clinical trial are shown in purple and yellow, and the trial was appropriately designed to distinguish the effect of dupilumab from other influences or factors. The development program also utilized FDA's recommendations to conduct anchor-based analyses to aid in the interpretation of the COA endpoint results. And these analyses incorporated the patient's perspectives to support that the DSQ score improvement in patients who received dupilumab in the clinical trial was representative of clinically meaningful improvement in dysphagia and therefore meaningful improvement in how these patients felt and functioned. This approval fulfilled the previous unmet medical need for therapies that were found to be safe and effective for these patients, and was directly related to the successful application of the principles of patient-focused drug development. Next. In closing, within the Division of Gastroenterology, as well as across other divisions and centers within the FDA, promoting the inclusion of robust, meaningful, and representative patient input has become a key element in clinical trial design and conduct that has advanced drug development for indications where the assessment of alternative endpoints are not practicable or feasible to conduct. The recent approval of dupilumab for the treatment of EOE highlights the importance and value of incorporating and assessing the patient experience in drug development, and would not have been possible without the implementation of key elements of patient-focused drug development discussed in PFDD Guidances 1 and 2. Thank you again for inviting me to participate in today's meeting and share a clinical regulatory perspective on the implementation and impact of PFDD.
Thank you, Dr. Lyons. Now we will begin session 3, our question and answer panel, to discuss what was heard during the meeting today. I will turn it over to Robin Bent to facilitate this portion of the meeting. At this time, all panelists should please turn their cameras on.
Great, thanks, Shannon. I know we're low on time, and so we are still trying to answer some of the questions in the Q&A chat. But let me start with one of the first questions that we received, which is, I'm interested in your insights about data collection methods among patients with disabilities. For instance, any movement or cognitive disabilities that might discourage them from engaging in completing surveys / questionnaires or interviews. And so I think it'd be really helpful to hear kind of our panelists' thought on that. It did come in during Dr. Noble and Dr. Daniel's presentations. So maybe let me turn to Dr. Noble to take this one.
Sure, thanks so much. And this is a really important question. You know, the consideration of impacts of physical and cognitive disabilities has to be factored in to considering the selection of research methods and study design with patients. And, you know, a one-size-fits-all research approach really may not be suited to the diversity of abilities that are seen within a specific patient population as well. And so I think that also needs to be considered. And so for whatever research methodology is selected, whether it's qualitative, quantitative, or mixed methods, we do recommend that you consider including patients and patient advocates as research partners to comment, like a patient steering committee, essentially, to comment on the method selection, the design, and the results interpretation as well, potentially. To pilot test your approach with patients to determine if the approach is patient-friendly and whether it's accessible for patients. You know, you want to consider patients who may be deaf or hard of hearing, patients who are blind or with low vision as well. To streamline and create brevity potentially in the interviews or surveys so that they're efficient and don't unnecessarily fatigue patients who may already be struggling with fatigue. And then to apply universal design principles to study documents and research tools. Universal design is the consideration of the design and composition of a study, and also the study tools, maybe questionnaires or web-based interfaces, so that it can be accessed, understood, and used to the greatest extent possible by people, inclusive of people with disabilities. Specifically speaking to patients with cognitive impairments, if patients can validly report for themselves, even if it's in a more limited capacity than cognitively typical individuals, consider using potentially supportive illustrations, which can be used to simplify ideas and provide visual aids to facilitate communication. Assistive technology. For example, assistive communication devices, especially if that's native to the patient and that's something they use in their daily life. And then also drawing activities or props potentially, which can also help elicit patient perspectives. But this is an important question, and creating inclusive research to make it accessible so that all patients can participate is essential. And thanks so much for the great question.
Thanks. I know that we're really very much coming up on time, but before, does anyone have anything they want to add to this question? All right, I know maybe we'll try and get maybe one more question in, and then for those of you whose questions we weren't able to address today, we will try to carry those forward to our upcoming meeting. But let me turn to a question that came in during Dr. Johnson's talk. How do you best manage representativeness in a rare disease where much of the recruitment is through an advocacy organization versus being able to find people through more traditional approaches? And before I turn to Dr. Johnson, I wanted to turn to Dr. King Kalamanis to see a lot of your talk focused on this, and I wanted to maybe ask if you had some initial thoughts.
Yes, and yeah, we see this request coming in for lung cancer as well. And so, I think really we do have to get a bit more creative with our language and how we talk to people and get the patients involved. If we have patient ad boards, ask them, would this resonate with you? And on your ad boards, don't try to, at least talk to a few different, groups of folks, not just the two, perhaps women who are on them. They tend to be female. Get more men involved in your research, get different perspectives, because what resonates will differ. So I think we can come up with a couple of different options for people to use outreach language, and that'll make a difference. But it's not easy.
I want to echo what was just said, and it was a great presentation for everybody. So thank you very much. But I do think that we have to break out of what may be traditional, and it can be uncomfortable, and you may need to be working with a lot of other organizations. And that is, I think we need to rethink our traditional recruitment paradigms, but that's true whether it's traditional advocacy organizations or other groups as well. So I'll stop there given the time.
Great, and my apologies to the panelists who stuck around for our Q&A session and who we're not going to get to hear from for a lot of these questions. But if you have any feedback, we're happy to share that maybe at our next meeting as well. With that, I'm going to turn back to Shannon because I know we're a minute over time just for her closing, and we will go on from there. Thank you.
Thank you all for submitting your questions, and thank you to our panelists for their contributions to the Q&A session. This is a reminder that the second meeting in the series will take place virtually on July 25th, 2022 from 11 a.m. to 1 p.m. Eastern Time. Next slide, please. Another reminder that this meeting and the second meeting have an associated public docket, and the docket is open to the public and will close on September 23rd, 2022. To submit a comment, please access the docket using the link on this slide and click comment. I would like to thank everyone involved in the planning of this meeting. I would also like to thank our presenters for their participation. Have a great afternoon.